Hello, welcome to the poster with titles Advance in the Early Diagnosis of Mucolipidosis Type 2. The small introduction of mucolipidosis type 2 is a very rare lysosomal disease caused by a deficiency of N-acetylglucosamine 1 phosphoryl transferase localized in the gene GMPTAB, which leads to impaired mannose 6-phosphate synthesis. This results in inadequate signaling of enzymes to lysosome, affecting multiple processes involved in the degradation of cellular components. Previously, diagnosis was only possible using molecular technique. This study demonstrates that early diagnosis is now possible using biochemical method, facilitating the early identification of this patient. Material and methods. A review of the clinical characteristics of a 4-patient diagnosed at our centers with this pathology using genetic method was conducted, and this study was expanded to include 5 patients, 4 external sources. Dry blood spot samples, DBS, were analyzed using multiple lysosome enzyme assay with 12 enzymes, and mass spectrometry was used for enzyme determination. These same samples were used for quantified proteomic analysis. The first resultant discussions. The study of 9 patients with ML2AB, 6 patients showing this table diagnosis with MLAB by whole exome sequences are included. All patients are recruited in different parts of Europe. The idea of use multiplex enzyme tests is look for one pattern is possible to detect the ML, in this case 2. In this table, we present the enzymes don't have any alteration, have value normal, or have a little alteration in one enzyme, in this case GBA, but don't is possible use with pattern for detecting the pathology in this case. The enzyme are normal in general is GALC, GLA, GBA, GANLS, and GLB1. In this table, we present the rest of enzyme, but it's possible follow one pattern because in all time are elevated in neonatal samples and the same patient with more older samples and with compare with other patient of different countries. All this is one case in page number 6, but at this moment is pending of the result of genetic test. The proteomic test complete with the samples previous show reveals one increment in the regulated proteins. Principal route we found regulated are the energy production pathway such as glycolysis, extracellular matrix, cholesterol metabolism, and other protein descompensates. If look for the enzymes have the pattern elevated all time in previous results, you can find all enzymes participate in 2 pathways, dermatan sulfate and heparan sulfate. As conclusion, the enzyme analysis studied relevated a pattern of very high enzyme levels. We can use the enzyme ASMD together with 1 or 2 enzymes participate in the degradation of heparan sulfate or dermatan sulfate pathway to obtain one early diagnosis to mucolipidosis type 2. Energy production pathways appear to be altered with increased enzyme activity in the dermatan and heparan degradation pathways. This is likely due to compensatory mechanisms to obtain energy from sugars, thought the degradation of the extracellular matrix and lysosomal glycogen, which will explain the elevated GGI levels. An alteration in cellular cholesterol trafficking could also contribute to the molecular basis of this pathology, which is currently poorly understood and studied in the relation to the MSMD elevated. Proteomic results point to an imbalance in these energy pathways. Some enzymes, such as GBA, are also beginning to increase, but it's not to use as a standard of detected this disease because they are not present in all the cases. Thank you for listening to this presentation. Thank you so much.